Thank you very much. It's a great pleasure and honor to be here participating at this meeting, and it's a great pleasure also to have benefited from the Studium support for this project, Coexistence near neutrality. So complexity and dynamics in ecosystems are current challenges in ecology. The questions of which forces shape ecosystems, how is biodiversity generated and maintained, and what are the fundamental principles for stability and resilience. These are not just scientific questions, but they are also questions that have to do with management of ecosystems, with challenges like climate change and human changes. So applications in the control and management of diseases, understanding the interplay between extrinsic and intrinsic factors, the balance of aggressive or moderate interventions, then phenomena that happen across multiple scales, deterministic forces and stochasticity, and responses to climate change. These are very challenging aspects of ecology that can be addressed with mathematical models and computational approaches. So there is a long-standing debate in ecology, the neutral versus niche debate for explaining coexistence and maintenance of biodiversity. And recent approaches are calling for a movement from the dichotomy between these two perspectives on coexistence to a reconciliation. So the niche theory postulates that species are fundamentally different and these differences allow them to coexist. Whereas neutral theory is based on the assumption and the perspective that all trophically similar individuals are functionally equivalent, and diversity results from a balance between speciation and extinction on continental scales, or immigration and extinction on local scales. So some of the questions that modern research is trying to address is what is the relative importance of niche and neutral forces in determining the composition of ecosystems? So trying to reconcile the two theories and finding ways in which they can be linked, and also looking at the role of transience and scale dependence in ecological dynamics and the manifestation of these forces. So why am I interested in biodiversity and the questions of coexistence? I work on infectious disease dynamics, ecology and evolution, and I'm very much interested in multi-type microbial ecosystems. In systems like dengue viruses, influenza, malaria, tuberculosis, pneumococcus, staphylococcus aureus, and SARS-CoV-2, the questions of what generates and shapes polymorphism are a very important public health issue. We have strains of these pathogens that differ in antigenicity, so we have antigenic variants that represent a different challenge to the immune system, antibiotic resistance variants that are targeted differently by antibiotics with different success, virulence variants that can cause disease at different intensity, growth and transmission traits can vary between strains, there are vaccine escape mutants, and many, many other levels of variation where you can think of traits that generate potential for ecology and evolution. So the questions are which variants coexist and why? What are the mechanisms? Typically, to answer this question, we need to construct high-dimensional systems to represent all this diversity with many feedbacks between the different compartmental classes and very complex models. We have to represent what happens in a single host and how that affects transmission at the population level. The analysis is often difficult, and typically we resort to simulations and cumbersome computation. So sometimes it's very hard to understand what is the backbone of the dynamics. But control depends on a quantitative mechanistic understanding. The better we understand the mechanisms, the better we can control such systems and also predict them. So endemic pathogens with multiple strains are examples, for example, include Streptococcus pneumonia, human papillomaviruses, Haemophilus influenza, and others. And modeling them is hard because they require representation of diversity that is always there. Typically, this is addressed with frameworks that are susceptible, infected susceptible, epidemiological models, but they are rare in the literature. Extended to 2 or more strains is considered very hard. And multi-strain SIS model with co-infection are even rarer. It has been studied theoretically that strains varying in transmission or clearance rate only can be studied easily through reproduction number hierarchies. And the strain with the highest reproduction number will win over the others. It leads to competitive exclusion. However, under co-infection, R0s alone do not determine the final outcome of competition or selection between strains. So far only low-dimensional models of co-infection have been considered and special cases. So the empirical questions that we'd like to address are why there is differences in variant prevalences and distribution across regions and also across time, and applications of such models in fields like antibiotic resistance, prevalence, evolution of virulence, or endemic prevalence in polymorphic microbial ecosystems will benefit from having models that address these aspects. Multidimensionality, so multiple strains, and co-infection. So this is the basis of our project, Coexistence Near Neutrality. So we seek to obtain methodological and also biological advances. The question is, how does collective coexistence arise from microscale, pairwise interactions and variability along multiple traits? We will consider co-colonization or co-infection as a route to coexistence. So think about a host that is colonized by strain 1, and then it can be co-colonized by strain 2. And then the transmission dynamics of these strains in a network of such encounters repeated over time will lead to epidemiological competition between these strains. So I will explain the basis of the model, but first, why is this important? This is, of course, a general phenomenon within and between species. It can be applied to pathogens and also commensal bacteria. There are many analogies with multi-species ecology and population structure, and it will help us to understand microbial type distribution and variation, the role of interventions, how do interventions like vaccines or antibiotics or even climate change actually change the dynamics of these strains. Understanding co-infection or co-colonization will also allow us to understand the potential for evolution via horizontal transfer and also virulence evolution. Of course, next generation vaccine formulations is a relevant topic, especially for Streptococcus pneumonia, which has many serotypes, and understanding the co-colonization patterns is important. So the basic model is the following. We have a system of hosts that are susceptible, infected by one strain, or infected twice. Co-infection classes are represented by the double indices 1, 1, 1, 2, 2, 2. So a host is susceptible, it can get infected by one strain, and the hosts that are infected by one strain can go to the co-colonization class. They clear infection at the same rate, so they go back to S without immunity, and hosts that are co-infected transmit either strain with equal probability. So in the beginning, we assume that the strains are completely identical except for the interaction in co-colonization. So when there are these transitions from a single infection to double infection, there is a potential for interaction between strains. The resident strain can inhibit or facilitate the co-colonization by the other strain. So when these coefficients are below 1, we have a competition or inhibition between I and J. When these coefficients are above 1, we have facilitation. So this model leads to 4 outcomes between 2 strains, either coexistence, exclusion, or bistability. And this all depends on the magnitudes and the distribution of these 4 coefficients. We have a 2 by 2 matrix for 2 strains. This was the beginning. And then when I met Sten some years ago, we started thinking about this in terms of timescale separation. So this was the first extension of the model where we looked at the dynamics assuming similarity in Kij. So everything else is the same, but because we are dealing with strains of the same species, we can assume that we can represent each of these coefficients or these interaction traits as a reference plus a deviation from that reference, where epsilon here is a small parameter. And this was the beginning of looking at the system in much simpler form, because what we find is that we can write the dynamics as two components, a fast dynamics, where the strains behave as neutral and only big variables stabilize, which is the total prevalence of susceptibles, the total prevalence of single colonization, and the total prevalence of double colonization. And on a slow time scale, which depends on this epsilon, the epsilon t time scale, under such global conservation law, the individual strain frequencies change. They follow explicit dynamics. So basically, we have two time scales, and these time scales are completely governed by the similarity structure between the strains. In this case, one parameter trait, Kij, that varies. 3 years later, we obtained an extension, a generalization of this framework to n strains. So this is not as easy as the two-strain system, because we now have an n by n matrix between, for interactions between a colonizing strain and a co-colonizing strain, which can inhibit or facilitate each other, okay? And which strain will win? That's the same question. What is the final dynamics? Which strains will coexist? Will they coexist? How stable will be the coexistence, and so on. So the model becomes more complex. We have the order of n squared equations, and still the idea is the same. Let's try to apply the slow-fast approximation, the similarity structure. So what we find is, again, it's very nice, because it preserves the same patterns that we found in the earlier work. So on a fast time scale, strains behave as neutral, where the total prevalence of susceptibles goes to its equilibrium, 1 over R0, the basic reproduction number of the strains. The carriage prevalence, the total, is 1 minus 1 over R0, 1 minus the susceptibles, and the distribution between single colonization and double colonization depends just on the two parameters, the mean field parameters, R0 and K, where K was this reference for the interaction, and R0 is basically the transmission intensity. It's beta over mortality and clearance rate, so it's the transmission intensity of the pathogen, the basic reproduction number. What happens on a slow time scale? What we find is that within such global conservation law, we have slow strain selection. And the selection is given by this n-dimensional system, where we have the frequencies of each strain, Zi, following this explicit equation. And this is very nice. This is n dimensions, because we have n species or frequencies of strains. And this is entirely determined by the pairwise invasion fitnesses between strains, where the pairwise invasion fitness is given by this expression. So it depends on these deviations from neutrality, on these alphas, the rescaled interaction strengths. And basically, we have how each strain will grow in the system. It will grow in the system if it's very good at invading other strains. So it's alpha I, lambda ij times zj. But also, it will suffer a cost from this global environmental feedback that comes from all the changing strain frequencies in the system. Okay? So this is basically a feedback that is created and is dynamic over time. But it's the same for all strains. So this is a kind of environmental feedback that goes back. If this summation here is positive, it's a negative feedback for all the strains. If it's negative, it will actually benefit all strains. And the key parameter here is also mu, which is I over d, the ratio of single to double colonization in the fast dynamics, in the global prevalence of the system, and is given by the two parameters, K and R0 minus 1. So for each pairs, we have 4 possibilities. So each edge can have 4 types. Okay? So if both lambda ij for 2 strains are positive, we have coexistence of the 2 strains. If they are negative, we have bistability. If one of them is positive, the one with the positive invasion fitness will win. But as we can see here, it's not just the signs of these that matter, it's actually the magnitudes. The magnitudes of the lambda ij decide the outcome of competition. And this equation here, it took us some time to realize that actually this is the replicator equation from evolutionary game theory. So this is very nice. It connects basically our system to a reduced version, to a reduced model, which is exactly the replicator dynamics. So how can we obtain the epidemiological variables? After this slow-fast approximation, what we have is that the single infection for any strain will be given by total single times the frequency, Zi, and the total prevalence of the hosts that carry I and J will be given by total prevalence of double infection or co-colonization, Zi, Zj. But this was not a priori, we didn't start with this, we actually derived this. So basically you can see that over the time, these products equalize, and the dominance of one strain in single colonization and double colonization also converge to the same value. So in this system, we have explicit dynamics of the frequencies, and then we can follow evolution because we can track which traits vary and how the mean of these traits vary. So we can see how the mean interaction changes in the system, the K effective, so it's K plus epsilon, and this quantity that changes over time, and it means that global cooperation can go up if this little q is positive, or global cooperation can go down if this little q is negative in the system, right? As frequencies change, the traits of the system change, right? And the q, which is this double sum of the lambda jk plus lambda kj times the products of all species in the system, this is nothing more but the mean invasion fitness, the lambda bar of the system. So this is a dynamic quantity which is actually the mean colonization resistance of the system, how easy it is for an outsider to invade the system. And it's very nice because we have a closed, completely closed ecosystem between the species, and we can follow how the invasiveness of each strain changes over time, and also the invasibility of each strain, how easy it is to be invaded by others. In this model, the strains can be seen as species. They are actually a subject that is evolving and has a fitness and is increasing in frequency, but at the same time is also the resource for other species or other strains in the model to grow via these co-colonization niches. Okay? So it's basically these two aspects of each strain that are changing over time. This is an example of the dynamics for n equals 6. So we have the Kij matrix. From this, we can derive the pairwise invasion fitness matrix. From the signs of the invasion fitnesses, we get this network, whether it's competitive exclusion between each pair or bistability or coexistence. And we apply the replicator equation, and it's very nice. We can predict explicitly the final dynamics of the system. So what we did in tour with a PhD student, we extended this model to multiple variation among strains. Until now, we considered all of them equivalent in all the traits. They vary only in the Kij. And with a PhD student, we say that the strains are more complex. There is variation along 5 dimensions. There is the Kij that is different, like before, but there are transmission rate differences for each strain. There is a clearance rate from single infection for each strain. There is a clearance rate from co-infection for each strain, and transmission biases from mixed co-infection. If a strain arrives first, maybe it has an advantage in transmission than when it arrives later in co-colonization. So basically, this makes the model more complex, the same dimensionality, but the strains are more difficult. So we have now these 5 dimensions. And again, the aim is, can we simplify the system? Can we get the same replicator equation? And the answer is yes. So it's very nice because we can again represent each system as for each pair, there is a combination of lambda ij. So for each pair, we have one of these 4 scenarios. But in the multiple strain system, this is a bit more complicated. So now, for each set of strains, the pairwise invasion fitness becomes a linear combination of all the variation among all the traits. Before, in the first version of the model, we only had the susceptibilities to co-infection. We only had this part in the lambda ij. Now, with 5 traits, we have basically how each trait contributes to the success, to the relative success of one strain over the other. And it's given by variation in transmission, variation in clearance rate, variation in co-infection clearance rate, and the priority effects. okay? And this is for the other strain. So basically, we can study very explicitly the dynamics of each pair of strains, which traits favor exclusion and which traits favor coexistence. And we can find also the weights, what are the weights of each of these traits, and study basically how the neutral model is actually affecting the selection between strains, because selection depends on the magnitude of the lambdas, and we know exactly these parameters from the neutral dynamics, from the global environmental dynamics of the system. So the replicator dynamics is the same, theta z, then lambda z minus this q quantity, the mean invasion fitness, but now the lambdas are a little bit different. Now the lambdas have to have this linear combination in them. So we can study the qualitative transitions of the system in detail. So by looking at the signs of the lambdas, we know exactly what is the outcome of the system. If the two lambdas are positive, there is coexistence. If one of them is positive, the other is negative, the strain with the positive lambda will win. So here you can see that as you vary this ratio of single infection to double, the signs of the lambdas do not change. So there is always the strain with the positive lambda that wins. Here, as we change mu, this ratio of single to double, which depends on these two mean field parameters, one of the strains wins in the first part and the other strain wins in the other. So the winner depends on the context. And if you have more traits varying in this particular case, you can have 3 transitions, 3 crosses. The signs cross 3 times. Now in this other way, if you have variation in co-infection susceptibilities, you can have coexistence or bistability. What does this mean? This means that the 2 curves are positive or negative at the same time. So you see here there is a region where, as you vary the ratio of single to double infection, the lambdas basically have these positive signs, and then only one strain wins for increasing ratio. So we can study this in detail, and also the frequencies at the coexistence level. It's exactly given by the ratio of lambda 1, 2 over the sum of the lambdas is explicit for 2 strains. So we can do basically a lot of analysis on the 2-strain system, where we find the regions of coexistence, the regions of exclusion, and how they depend on the interplay between the trait means and the variation. Because these curves here, the blue in this inequality, how big the variation has to be for coexistence to be maintained. So the variation has to be small enough for the coexistence to happen. Okay? And these limits here are actually determined by the mean of these parameters. So it's really very nice. And we can study in the 2-strain system also 4 transitions. So I will draw your attention here. This is the system with 2 strains, but now we have this 5-dimensional variation. So we start here and we increase this mu parameter. And what you can see is that the same system actually crosses all the 4 quadrants. So it goes from exclusion of one strain to coexistence, to exclusion of the other, to bistability, back to exclusion. It crosses 4 times. And here you can see the lambdas, how the lambda functions change as a function of mu. And what we need is that to have 4 transitions, we must have that the strains vary in clearance rates of co-infection and also vulnerabilities to co-infection, Kij. Okay? We can study also the role of how you change mean field parameters, and then you have non-monotonic changes in the frequencies. So here I'm changing the clearance rate on the bottom axis, and I'm looking at the focal strain frequency for different assumptions of the variation between the strains. And what you can see is that sometimes you can have non-monotonic increases or changes in the frequency. So you increase the clearance rate of everybody for some time. You see the focal strain frequency goes down, but if you increase the clearance rate further, the focal strain frequency can go up. So it's non-monotonic. So clearance rate, for example, the global clearance rate can be changed by increasing the antibiotic treatment of the whole population. So the mean field changes can have different counterintuitive effect on a focal strain frequency. And this depends on the details of their variation. But in this model, it becomes completely explicit. So I will conclude by this small digression. I'll go back to the simpler model that we started in the beginning, where we had just variation in the Kij, because that's actually a very strong modulator of coexistence. So in that first model, when we see the lambda, the pairwise invasion fitness depends on alpha ji minus alpha jj. Then you have this negative sum here, alpha ij minus alpha ji. This measures the antisymmetry. How antisymmetric is this alpha matrix? So basically, the bigger the bigger the mu is, the more important will be the asymmetry in the alphas. The more it will contribute to the lambda ij. And you can see if you generate the alphas randomly from a distribution with mean zero normal distribution, you will see that the relative proportion of the pairs that are in coexistence, bistability, exclusion of one, exclusion of the other, will be roughly equal, okay? But as you increase mu, this parameter, for the same distribution of the alphas, you will see that the strains tend to have negative lambda ij. So one of them will be negative, the other will be positive. So they will tend to pairwise exclusion, okay? And this leads to complex coexistence. So what you can see, the effect of this is that as you increase this mu parameter, which for example can be changed by R0, if you go from high transmission to low transmission, very different coexistence dynamics emerges. So you go from stable coexistence to less stable, oscillatory, then you have limit cycles and these sort of heteroclinic attractors where some strains exist for a long time alone and then are replaced by others. And you can study this also more generally to see that how important this ratio of single to co-infection is in the system by studying what's the probability of finding the system in a multi-stable regime, a single stable regime, monostability, what we call here, or a complex regime where you find no steady state at all. So this is the complexity regime, what we call. So this is very, very interesting. So we see that as you increase this ratio, you have more complex coexistence. And there are two ways in which you can change that ratio mu. You can change it by K or R0. If K goes up or R0 goes up, you are in the regime of global facilitation or high transmission. And what is the dynamics is typically this multi-stable coexistence. A few strains coexist and co-colonization dominates. When you decrease K or decrease R0, you go to the regime of global competition between everybody and low transmission prevalence. And in this case, single colonization dominates and the dynamics become very complex, where you can see that also system properties, system phenotypes like this mean invisibility, the red line here, is oscillating because the strain frequencies are oscillating, so the system properties are also oscillating. Whereas here, you have a stable regime, a stable coexistence between strains, which means that the phenotype of the system is also more stable. What is the only way to preserve mu? If you think, we have a very simple expression. Mu is a very critical parameter in the model. It's I over D, the ratio of single to double infection in the neutral model. And is given by this ratio between 1 over R0 minus 1 and K. The only way to maintain it constant is if you trade off R0 and K against each other. We see that if you change mu, you change the stability of the system, you change the number of species that coexist. It's very sensitive. So if you want to preserve a coexistence regime, you should preserve mu. And this means if the system has a higher basic reproduction number, if you increase transmission, you can compete more. The species can increase competition. So basically, the K goes down. If R0 goes down, K goes up. So this line here is the same mu, so it means you preserve the same qualitative coexistence between the species, but of course, it's a different, slightly different environmental regime. The speed of dynamics can be different because the theta in the replicator depends on R0 and K. So basically, it's a way to connect our model naturally to the stress gradient hypothesis. In ecology, the stress gradient hypothesis postulates that if the environment gets more hostile, species cooperate more, and if the environment gets more favorable, species compete more. And this is what we have here, explicitly emerging naturally from the dynamics and the sensitivity of the system to mu. Now, it is important to study what kind of trade-off will hold in the full model with variation along 5 traits. So this is something that we have to study in the future. So in summary, we have found a new path for full analytical understanding of SIS dynamics with co-infection and multiple strains, and also that vary now along multiple traits. We link neutral and non-neutral dynamics by explicit feedbacks and results, and we propose timescale separation as an approach to enhance insights for similar strain dynamics. This can be applied to study speciation, evolution, parameter inference. Replicator dynamics is surprisingly little found in epidemiological models, and even little used in ecology, because most ecological models are based on the Lotka-Volterra system, but it's not completely equivalent. We consider pairwise invasion fitnesses as driving forces in the n-player system, so it's very important to study these quantities. And there are many plans for the future. We're looking at critical transitions from the edges of the network to the full dynamics, finding links with data, both from epidemiological and microbiological systems is important, and theoretical extensions, like including space, thinking about system assembly, sequential increase of species over time in the system, and optimization. If we want to optimize any quantities in the systems is important. So I would like to conclude by thanking my main collaborator in this project since 2016, Sten Madec from the Institut Denis Poisson, University of Tours in France. And this work has begun at IGC some years ago, where I was at the Gulbenkian Institute. Now it's Instituto Superior Técnico, and was also very much facilitated by the Studium visiting researcher in 2021. Thank you very much for your attention.